Welcome back to campus. For those of you I haven't seen yet, but my name is Marianne Hunt. I work in the Alumni Relations Department in the lifelong learning area. And in our department, we work to bring you educational content, programming like this, faculty lectures at reunion and at homecoming and other home football games. We also go around the country with our Dartmouth on Location program, and we even go abroad with our alumni travel program, which I'm very pleased to say that both of our gentlemen who are speaking this afternoon are willing to participate in as well. So if you want to learn any more about those programs, please see our website, alumni.dartmouth.edu. And I would like to take a moment to just remind you, because there was a little bit of noise when that announcement was going on, to turn your cell phones down, please. And it is an extreme honor for me today to introduce you to Professor Nate Dominy and Jerry DeSilva. I've had the pleasure of working with them on many of our programs, and as I said, happy to be working with them on more coming up. And you'll have to forgive me now because I'm going to read their credentials because I don't want to sell them short, but they are so qualified that it's going to require a little bit of reading. So Jerry DeSilva is an assistant professor of anthropology. He attended Cornell University for his undergraduate and received his PhD from the University of Michigan. He is a paleoanthropologist specializing in the locomotion of the first apes and early human ancestors. His particular anatomical expertise, the human foot and ankle, has contributed to our understanding of the origins and evolution of upright walking in the human lineage. He has studied wild chimpanzees in western Uganda and early human fossils in museums throughout Eastern and South Africa. Professor Nate Dominy is an anthropologist and evolutionary biologist. He studies the behavior, ecology, and functional morphology of human and non-human primates. He has worked in Africa since 1999, receiving grants or fellowships from the National Institute of Health, National Science Foundation, National Geographic Society, Packard Foundation, Mellon Foundation, and Smithsonian Tropical Research Institute. He is an elected fellow of the American Association for Advancement of Science, the Explorers Club, and the Royal Geographic Society. So you can see why I needed to read that now. His research philosophy is to integrate tropical fieldwork with mechanical, molecular, and isotopic analysis in order to better understand how and why adaptive shifts occurred during the primate evolution. And today they're going to tell us about their exciting research and what they're doing with our undergraduates. So please help me welcome Nate and Jerry.
Okay, thank you very much, Marianne, for that very kind introduction. I am delighted to be here to welcome you back to Dartmouth and to introduce myself and Jerry DeSilva and talk to you a little bit about our own research interests and how we've chosen to integrate our research with our educational interests. So I will begin by introducing ourselves a little bit, and I'm going to turn over the stage to Professor DeSilva to talk about his own research, then I'll talk about my research, and then jointly we'll talk about a recent course that occurred on Dartmouth and in South Africa during the last fall quarter. So here we are at a fossiliferous site in South Africa, and Professor DeSilva arrived in Dartmouth in 2015. I arrived in 2010, and we realized that this was an exciting opportunity to integrate our research and our teaching interests because we have shared interests in South Africa. Jerry studies early human ancestors, fossil hominins, we say, and how they moved about their landscape. I'm interested in feeding. I think food is pretty important. After all, you have to eat to survive. And I think most of the milestones of human evolution can be interpreted some way through the lens of foraging behavior. The way that humans detect food on the landscape, the way they engage their anatomy to acquire that food resource, and the way they engage their digestive system to assimilate resources from that particular food item. I think all of those stages are important, and South Africa is a wonderful place to study those kinds of interactions. And so together we realized that his interest in the fossil record, the deep time history of South Africa, my interest in contemporary foragers, humans and non-human primates, and how they interact with that landscape, that this was a really perfect storm of opportunity and interest to create a really exciting course. And so this slide is meant to transition to our particular research interests. Here is an example of a fossil hominin that's relatively complete. We'll learn more about it. We actually have a cast in front for you to see. And the way this thing walked, we think, is important for instructive for examining its paleobiology, the way it interacted with this landscape. And here I'm focused on contemporary humans, modern humans that are also foraging on this landscape. And the way they solve this ecological problem may help us understand how early human ancestors solved similar ecological problems. So with that, I will turn it over to you, Jerry.
Great,
Yeah.
thanks, Nate.
Okay.
And welcome back, everyone. It's really a pleasure to be here to talk to you about my own research and to tell you a little bit about how Nate introduced our role in this class, Anthropology 70, Experiencing Human Origins, and how our research integrated beautifully with one another and with this opportunity to teach 15 students in the fall term of 2016. It was really an extraordinary experience. Now, as Nate mentioned, as Marianne introduced, I'm interested in the origins and evolution of humans. We know by studying the genetics of humans and our closest ape relatives that humans and chimpanzees are first cousins of one another, and that they share a common ancestor that lived about 7 million years ago, presumably on the African continent. What we don't know in as much detail is the path and the process by which humans became human, and that's where we rely on the fossil record. But the fossil record, even though we have thousands of fossils, we're talking about millions of years. And so we only have a couple of fossils representing each few thousands of years of existence. So every time we find new fossils, it really does help us complete the picture that much more. And oftentimes it forces us to ask questions we never even thought to ask in the first place. So discovering new fossils is really, really, really important in understanding why humans are the way they are. Now, the particular group of early human ancestors that I'm interested in are a group called Australopithecus. Many of you probably know about Lucy, the famous Lucy skeleton discovered in Ethiopia in the 1970s by Don Johanson. She's an Australopithecus. But we find lots of different kinds of Australopithecus throughout Eastern and South Africa. And in fact, the very first Australopithecus fossil ever discovered was found in South Africa in 1924 by an anatomist by the name of Raymond Dart. This is known as the Taung child. Raymond Dart was interested in these fossils that were being discovered from limestone caves. They were being blown out of limestone caves by the miners, and he was interested in the baboon fossils that were being found. Well, one day, and as the story goes, who knows how true this is, but the story is he was in a tuxedo preparing for his best friend's wedding, and a box arrived at his door. And he opened up the box, and this little face was staring at him. And he went to the wedding, and he came back quickly and pulled out his wife's knitting needles and slowly etched away at the rock and freed up this absolutely beautiful little skull of a child, what we know now is a roughly 3-year-old individual. But it was no monkey, or it wasn't even the anatomy that we see in modern apes. In fact, what he noticed were the small canine teeth, which is more similar to what you find in humans today. And he noticed that although the brain, the preserved fossilized endocast of the brain, was small, like an ape's would be, it was structured more like a human's. And the hole at the bottom of the skull would suggest that this thing could walk around on 2 legs, like we do. And that's my particular area of interest, is the origins and evolution of upright walking. Now, since that time in the 1920s, it's been almost 90 years, and researchers have found many, many, many fossils in South Africa. In fact, the region right outside of Johannesburg, where we went with our students, is known as the cradle of humankind. Now, that doesn't mean that's the only place where humans were evolving, but it's littered with cave sites where we find a really nice record of human evolution. Now, after Raymond Dart worked on the Taung child, fossils were found at a site called Sterkfontein, which we'll talk about later. Fossils were found all through this valley. And there's a colleague and a friend of mine by the name of Lee Berger who worked at the site of Gladysville, which is shown up there in the upper left. And he worked there for about 20 years. And at Gladysville, he found about 20,000 fossils. This sounds amazing, right? 20,000 fossils. What he found were antelopes and wildebeests and warthogs and giraffes and elephants and zebras and baboons, ancestors of those. He only found 2 fossils of early humans, a tooth and a pinky bone. That's it. Early human fossils are exceptionally rare. They are very, very difficult to find. Now, this was frustrating to him. And so he embarked on a new strategy to find fossils. Instead of going back to the same sites over and over again, let's find new sites. And what he took advantage of was this new technology that was available to the world at the time called Google Earth. And anyone who's used Google Earth, one of the first things you do is you look onto your own house. You look at a landscape that you know very well. Well, he knew this landscape well, and he decided to dive into this landscape using Google Earth. So here's an animation where we fly into South Africa, into the cradle of humankind. And when we get there, it's pretty brown. It's pretty dry, arid landscape, tall grassland environment. But every once in a while on this landscape, there's going to be a cluster of trees that's illustrated with these red stars. And the question that he and his geology team was asking were, where were these trees? They were mostly olive trees and stinkwood trees. Where were these trees getting their water? How were they getting enough water to grow? And what they figured out was that these trees were growing out of vertical cave shafts, that water was pooling at the base of these caves, seeds would come blowing in, and they would sprout out of the caves, meaning that the tree clusters were acting as bullseyes for where there might be additional cave sites, where there might be additional fossils. And using this approach, he discovered close to 500 new caves on a very well-explored landscape that nobody had known about before. And one of these caves, the one that the animation is circling around right now, is known as Malapa. And that's where we took our students, and that's where I do a lot of my research. Now, from the sky, Malapa doesn't look very impressive. It just looks like a cluster of trees. And from the ground, it looks equally unimpressive. It's just a hole in the ground. There's just a pit there. This is the kind of place I probably would have walked right past, expecting fossils to be preserved in a much more extraordinary landscape or within a cave that was just beautiful. And this isn't, it's just a pit in the ground. And yet that pit yielded two of the most complete early human skeletons, including this one here, that we've ever discovered in the history of our science. But what I love about this discovery story is that the very first fossil wasn't discovered by the paleoanthropologist Lee Berger. I'll let the discoverer himself tell you what happened. Matthew Berger, Lee Berger's 9-year-old kid, found the first fossil of what has turned into one of the most extraordinary discoveries in the history of our science. It is a wonderful, wonderful discovery story that loops in the public really well into our science and certainly gets the students really excited about fossil discovery, because if a 9-year-old can do it, then anyone can, right? And so, right place, right time for Matthew. But one of the questions that's often asked is how did they know this was an early human just based on a clavicle or a collarbone? Well, Lee Berger had studied that shoulder girdle for his dissertation. He knew this area of anatomy very, very well, but still there might have been doubts. And when they flipped over the rock to the other side, there was the lower jaw sticking out with a canine tooth. And remember, the canine is small and blunt in humans, and it's sharp and pointy in some of the other primates. And so they knew right away that they had made a really extraordinary discovery. And within 5 minutes of being at Malapa, Matthew Berger, the 9-year-old, had made as many fossil discoveries as his dad had made at Gladysville in 20 years. So a wonderful, wonderful discovery by Matthew. But that wasn't the end of it. As we started excavating this site, more and more fossils were revealed. They were all stuck in chunks of rock. I have one of these chunks of rock here that folks are welcome to look at afterwards. Fossils are rarely found just lying there, Indiana Jones style. They're found stuck within chunks of rock. And these chunks of rock have to go back to the laboratory, where under very, very careful eye, usually through a microscope and a steady hand, researchers will slowly, grain by grain, use a tiny little mini jackhammer to relieve the fossil from its surrounding matrix and free up those fossils. So here's an example of a chunk of rock that has a fossil sticking out of it. That's an upper arm bone, that's the humerus. And to the far right, it's sort of broken abruptly. That's actually an open growth plate. So we could tell from that bone that this was from a child, this was from a young individual. Now, a careful eye might spot in the upper right-hand corner, I'm trying to point it out there, a tooth. Oh, thanks, Nate. So there's a tooth sticking out of that chunk of rock. Now, when you see a tooth, oftentimes you're hopeful that there might be more there. Sometimes there's not. Sometimes there's just an isolated tooth. And you could spend months and months and months and months hacking away at that rock with your tiny little mini jackhammer and find nothing. So it sure would be nice if we could speed up the process, and that's exactly what happened. This chunk of rock was snuck out of the laboratory in the middle of the night and brought to the hospital. And at the hospital, Lieberger's wife, Jackie, happens to be a radiologist, and she gave it an x-ray. And when she gave it an x-ray, this is the image that appeared. There is a skull in there. That's the tooth sticking out right over there. Thanks, Nate. And then the silhouette of the skull is pretty clear from the side. Now, with more information, our preparators knew exactly where to hack away at the rock to reveal what turned out to be one of the most complete early human skulls ever discovered. It's absolutely magnificently preserved. Usually when we find fossilized skulls, they're in pieces and we have to glue them back together again. In this one, no gluing required whatsoever. This was in one piece, an absolutely beautiful, beautiful specimen. But a specimen of what? What is this thing? And as more and more of these rocks were prepared, we revealed not just a skull or a clavicle and a jaw, but two partial skeletons. On the left, we think is an adult female. Female because of her size and the size of her teeth, but she is an adult. She has wisdom teeth erupted already, and they're pretty worn down. And on the right, we think is a juvenile male, who would have been about 8 to 10 years old when he died based on his tooth eruption patterns and his growth plate fusion patterns. These two individuals are not the only ones we found at the cave. I'll tell you about others in just a second. But head to toe, their anatomy was different from anything we'd ever seen before. They were an Australopithecus. They were like Lucy in many, many ways, but they were different in other ways, and different in a combination of ways that was entirely unexpected. Their heads, for instance, were small. Their brains were small, but they were organized a lot like a modern human brain. Their arms were long, and their shoulders were shrugged upright like an ape's, suggesting they would have been quite comfortable in the trees. And yet, at the end of those long arms was an amazing human-like hand with a long, robust thumb that would have been capable of precision grip. Most of my research on these skeletons had to do with the lower limb, and the lower limb of a species that we called Australopithecus sediba. It was named a new species in 2010, and that's around when I started working with this team on this project. From the hips down, the hips and the knees and the ankles and the feet were indicative of something that walked on two legs, but differently than anything I'd ever seen before. It walked differently than we do. It walked differently than an upright chimpanzee would. And it walked differently than Lucy would have walked. And so we studied this. I worked with a physical therapy team, and we studied exactly how this skeleton would have walked around its world and why it would have walked the way it did. We think that because it was really good at climbing trees, when it did come down from the trees, that would have impacted the mechanics of its gait when it was on the ground. And we think that its bones tell us a story of an animal that walked with a gait that we call hyperpronation. And we published this in the journal Science in 2011 and again in 2013, our analysis of the foot and ankle and knee and hip of this creature and how they would have worked together to help us understand how this creature walked. Now, I can tell you with words how this happened, or I can work with a really talented Dartmouth student who can visualize this for us. There's a woman at Dartmouth who just graduated named Amy Zhang, and Amy works with the Dali lab, where their interest is in combining art and science. And Amy has been working with me for 1.5 years now to bring that skeleton back to life. After 2 million years, how did she walk? And Amy has now prototyped using Hollywood animation software, using what Pixar uses in order to animate their movies. She's now getting Australopithecus sediba walking after 2 million years of being in the ground. This thing is now walking again. It walks a lot like we do, but there are subtle differences as well, and those differences are going to help us understand the evolution of bipedalism. What we're finding is that there must have been these different experiments of bipedalism happening at this time period in human evolution. There wasn't one way to walk, there were all these different ways to walk around the world. Now, the last thing that I'll mention about this site is that we found more remains. It's not just these two individuals. It appears as though we have remains of an infant. There's an elbow from an infant, there's a shin bone from an adult, and there's an ankle from a toddler. So we have an infant, a toddler, a preteen, and 2 adults, which looks very much like this may be a family group. But we've got lots and lots of questions and lots of fossils still to discover. And in fact, what you'll hear about a little bit later is that when we took students to Malapa, they did make a discovery of one of these fossils. So with that as an introduction into my research and what I do and the really cool stuff that I get to work on, I'm going to turn it over to Professor Dominy to talk about his research as well.
Okay, thank you, Jerry. So I want to now move us from the lower limb up to the skull, and particularly the teeth. So one of the curious aspects about hominin teeth is that the relief on the teeth is relatively low. So if you run your own tongue over the surface of your chewing teeth, your molar teeth, you'll note that it's relatively flat. There's some relief, minimal relief to the tooth surface, and the enamel is relatively thick compared to other non-human primates. And so it's long been argued that the thick enamel of the teeth of our human ancestors and our own teeth is an adaptation to eating something relatively hard or difficult to chew. And this assumption was bolstered by the discovery of another type of hominin species in South Africa at the time. And this thing is called Paranthropus robustus. And I'll just note the flaring cheekbones here, and those cheekbones would have served as attachment points for the masseter muscle, this muscle on the side of the face that when you really clench your teeth, you can feel that muscle just pop out. That muscle is important. If you're chewing something really difficult, maybe like pizza crust or something like that, you can really feel that muscle working hard to chew. These animals, we can infer, had a massive masseter muscle. In addition, you'll see that they have this little crest at the top of the head. That serves as the attachment point for another muscle on the side of the head. So if you clench your teeth, you can feel another muscle up here. That's called the temporalis muscle. It's responsible for closing your jaw, so it also contributes to chewing. And that muscle passes through this hole here called the zygomatic foramen. And because the force of a muscle is proportional to its cross-sectional area, we can actually calculate the bite force of this animal, and it was immense. It would have had a bite force that was equal to or greater than a gorilla's bite force, but this thing was about 3 feet tall. So imagine something 3 feet tall with massive chewing muscles capable of chewing something very difficult to chew. And so these observations of these large chewing muscles has led to a variety of different hypotheses, the first of which was called the osteodontocratic hypothesis, which is sort of a mouthful, but it argued that these early human ancestors were probably using bone tools or stone tools, and that they were moving bipedally across the landscape, and they may have been maiming and clubbing small animals and then eating their flesh. This was an idea that sort of argued that early human ancestors were prominent hunters, and this is an idea that prevails in the discipline today, that the shift from an ape-like ancestry to a bipedal human ancestry may have involved some important dietary shift, and maybe the greater inclusion of meat was a central factor. But the mechanics of those teeth with the thick enamel and the low relief and the big chewing muscles has led paleoanthropologists over the years to conclude that whatever they ate, it must have been hard. And so here are some descriptions of the hominin diet. It was tough and abrasive, hard foods, hard and brittle, something very hard, a variably tough diet, hard, tough foods, so on and so forth. So we've been stuck with these mechanical descriptions of the diet, but we've been struggling to identify what those objects were. And that's been an important area of my own research. And here, the power of comparative biology may be useful. This is a map of the teeth of ourselves, humans, chimpanzees, gorillas, and orangutans. And this three-dimensional map is showing you enamel thickness. So the warmer the color, the redder the area, the thicker the enamel. So we humans have incredibly thick enamel compared to chimpanzees, gorillas, and to some extent orangutans. Now, that is probably an anachronism. We're not consuming hard things now. We spend a lot of time processing food, cooking it mainly to soften it. So we really don't need this thick enamel. So it's a bit of a puzzle. Why do we have thick enamel, and why do our ancestors have thick enamel? Chimpanzees, remember, 90% of their diet is ripe fruit, something very soft. So this is the kind of tooth you'd expect for consuming ripe fruit. So there was a shift. There was some shift from ripe fruit eating, we think, to something very hard. Now, orangutans may be informative. Look, their molar tooth looks rather similar to ours. And through the 1960s and 1970s, orangutans were considered the sister taxon to humans, and it was widely viewed that humans may have had, their origin story may have occurred somewhere in Southeast Asia, where orangutans live. Now, of course, with the molecular revolution, we know that chimpanzees are the sister taxon to humans, and the fossil record verifies that human origins happened in Africa. So there's a puzzle here. What dietary shift could account for that change in human origins? And the other puzzle comes with the chemical signature. These teeth that we see in Australopithecus sediba and others of her ilk is that they have this C4 signal, this chemical signal that suggests that a lot of their food is somehow derived from grass, which uses C4 photosynthesis. Now, it could mean that these hominins were eating grass directly, they were grazing, and that seems improbable. Animals that eat grass don't have relatively large brains. Or these animals were consuming the animals that consume grass, and they were hypercarnivores. But then again, those teeth are not well suited for chewing meat. Our teeth are just way too flat for efficiently processing meat. In fact, back of the envelope calculations of chimpanzees, occasionally they'll kill monkeys and they'll chew the meat. They spend more energy acquiring the monkey and then chewing it than they actually get from the meat itself. So for an organism that can't cook, there is a negative energetic benefit to consuming meat. And we think the main reason why chimpanzees might consume meat is for solidarity, group cohesion maybe, or blood is pretty salty. There's not a lot of salt in their environment, so maybe they just like the taste of salty blood. We're not entirely sure. But one idea for something that could be both hard, physically hard, that could explain the morphology of the teeth, and also have a C4 signature to explain the chemical composition of the teeth, are these underground storage organs. Now, South Africa tends to be, has a rich biological diversity of plants that have these underground storage organs. Globally, the Mediterranean has a lot, California comes in second, and South Africa comes in third, and Western Australia comes in fourth. So Mediterranean climates, Mediterranean habitats tend to favor this kind of growth form, and South Africa is relatively rich. And here's some photographs of the different kinds of forms. So this is an example of a corm, where botanically the stem is enlarging and swelling underground. So it's like a safety deposit box. The plant is putting its resources, its sugars underground to protect it during times that are unfavorable for growth. This is a corm here. This is a bulb here. And a bulb is just simply a leaf that's put underground. So it's a modified leaf. So when you consume an onion and you slice through the onion and you see those concentric layers, those are actually swollen leaves that have been put underground and wrapped around each other. Tubers are another form of underground storage organ. So a carrot, for example, is an underground storage organ. And a potato is actually a swollen rhizome. It's an underground root that connects two plants that are identical. So there's multiple types of underground storage organ, but they all share the same function for the plant, and that is to store resources underground. And so provided you're smart enough, provided you can move on the landscape bipedally and identify a particular species, and you understand that there's something underground, you have a big advantage over other organisms on the landscape that might be also looking for sugary resources. Now, there is another competitor that lives in South Africa. This thing is called the mole rat. Now, this is part of my research. I love mole rats. They're neither moles nor rats. They're bathyergids. They're more closely related to guinea pigs and porcupines than they are to moles and rats. These animals are eusocial. There's a single reproductive female. All the other females suppress their reproductive interests in the service of a colony. These animals are basically blind. They use seismic communication. They patter their feet to communicate with other individuals. There was recently a Nature paper that showed that some animals don't work as hard as others, and they get unusually fat. And that seems unfair. That animal's violating a social contract. It's not contributing to foraging for the group. And it turns out that those animals are the dispersers. So it seems like every other member of the group recognizes that that individual has a tough life ahead of it, and its chances of survival are pretty low. And so they allow it to get fat. And then when the rains come and the ground gets softer, it tunnels its way off to who knows. And maybe it'll be lucky, maybe not. In any case, these animals live in South Africa, and they consume these underground storage organs exclusively. This is the only thing that they eat. And here's an example. This is what South Africans call a volcano, and it's where the animals ejected dirt from its tunnel out onto the surface. And if you go across the South African landscape, you can see these volcanoes, and usually right next to one is a tuber or some other plant with an underground storage organ. And this one has actually been consumed a bit by a mole rat. You can see where the flesh has been excavated. And these animals, it turns out, are very courteous foragers. They'll take soil and they'll patch in the wound of the plant. They won't kill it. They want to save it for later. So they're like sustainable harvesters. Super smart. They're also magnetoreceptive. They're the only mammal that's known to detect magnetic fields and orient towards magnetism. So just super, super cool animals. I love this photo. It looks like it's got like a victory sign here or something. I don't know. It's pretty neat. This is a naked mole rat, so they're sort of famous. Cornell has a colony, and they learned quickly that you cannot keep them in concrete containers because they can chisel through the concrete, so they now have to use Pyrex, so pretty neat. In any case, there's a lot of mole rats across South Africa, so here's a map of South Africa, and each of these colored symbols represents an area where we sampled some mole rats, in addition to mole rats from the same fossil localities where some of these hominin ancestors were found. And so we did some research to look at the stable isotope composition of these different species. So here are the two hominin species, Paranthropus and Australopithecus, and this shows their values. These mole rats over here are a far away isotopically from these animals, and they're the tuber specialists. This animal, it comes from Cape Town, and it eats the rhizomes of what we in the United States call Bermuda grass. And then this animal is the corm and bulb specialist, and it has an isotopic signature that overlaps those of the two hominins. And so I think these data are pretty cool because they show that you can consume these underground storage organs and get the same isotopic signature as the two hominin species. And so I think these underground storage organs are therefore pretty viable foods for these hominins. These data come from fossil mole rats that were found in the same assemblage as those hominins. And again, those stable isotope data overlap the hominins. So you don't need to eat meat, which was the original explanation for that chemical signature. You can get the same chemical signature from eating plant foods. Now, humans have evolved the capacity to use fire, and fire is one of the hallmarks of human evolution. It's a major milestone because we think humans could then use fire to reduce the difficulty of chewing foods, and that the use of fire may explain why our jaws got so much smaller, why our teeth got so much smaller, because we can now chew things to a much lesser extent, and that gives us free time to do other things, and then brain expansion may be associated with that. So we can work with modern people who are consuming tubers and other kinds of underground storage organs, and we can study the effects of cooking. And this is a project, this is a project that sort of confirms the obvious because we cook underground storage organs all the time. We cook tubers, we bake potatoes, for example, and we already know that cooking them softens their properties. And this, we verified this with field data. So we think that one of the major reasons to evolve controlled fire use is to cook these plant resources. And this will come back, this theme will come back a bit later when we talk about the cores. Now, an additional aspect of South Africa is that you get these bored stones. So it's a piece of granite or schist where a hole has been bored through the middle. And these bored stones are common across South Africa. They're also found in North America. So the only two places in the world where indigenous populations would bore stones are in South Africa and North America. Now, in North America, we think they were mainly fishing weights. In South Africa, it's been a bit of a mystery. And it's been argued that the San, an indigenous group of people that live in southern Africa, were never indigenous to South Africa. This is a convenient argument for the current Bantu population living in South Africa. But this discovery that was published a few years ago, where they found a bored stone dating to 40,000 years ago in Border Cave, does a lot to dispute that argument. And these bored stones are everywhere. Farmers in South Africa regularly find them in their fields, and they bring them home, and they use them mainly as doorstops. And so I've done some work in South Africa where I've worked with farmers, and I collect their bored stones, and I measure their mass and everything. And here's one that was only partially bored. Here's a sense of what it looks like on the inside, and that is granite. That would have taken a lot of work to bore that stone. Now, it's been argued that these bored stones were mainly used to add weight to a digging stick. So here's an image of rock art showing a woman with her collecting bag. Here's a stick with a stone on the stick, and here's a plant with an underground storage organ. And here's a photograph from the 1880s. It's a stage photograph showing a woman with this bored stone on her stick. And here's another stage photograph from the 1980s. So we've never actually directly observed people using these bored stones. It's just assumed, based on the rock art and the nature of the stick, that it was advantageous for digging. Now consider this. These women weighed about 100 pounds. The average weight of the stone is about 3 pounds. And these women, we think, moved about 18 kilometers a day in the search for food. So that is a non-trivial mass to lug around the landscape with you. So there must be some advantage, mechanical advantage to using these stones. Here's another image of rock art from the area where we did our work. It's in the southern coast of South Africa. And it shows some women on a collecting party, we think, with the stick and the stone. And there's a little plug to keep the stone from moving downwards. And so that's a clue that, again, another clue to suggest that the stone was meant to weight a digging stick. We actually used board stones from the archaeological record from this area. So these are actual board stones that were found in that same area where that rock art was found. And in those same archaeological sites, you see these rich beds of this botanical tissue. Now, this botanical tissue comes from an iris. So the irises that you plant at home come from South Africa, and there is a tunic around that cormus tissue. And this is the tissue that you'd remove from the corm to consume it, and so this is refuse in the archaeological site. So we can do an experiment where we create a stick. Here's a stone. We know the mass of the stick. We know the mass of the stone, and these little tapes tell us the distance between segments. So there are increments on the stick and the stone. And we can work with women in the same landscape, and we can measure how much force is generated every time the stick strikes the soil, because through film analysis, because we know the speed of the stick and because we know the mass of the stick and the stone and everything, and we can calculate the weight of gravity, we can estimate how much force and translational momentum was put into the ground. And so this is just, I'll just mute this. You just see what it's like. So the target plant is right here. And these plants grow right next to large rocks, probably to protect, they're not conscious of this, but mole rats are on this landscape. So the closer it is to a large rock, the safer the plant is. And you see the attitude of the stick is constantly changing. That's why we needed the tape on the stick, because that distance will never change. So in our film analysis, we always know how fast it's moving. So the velocity and the force, so the velocity and the mass will give us an estimate of the force. All right. And then this is the final outcome. This is the corm itself. This is the edible tissue. It's about 80% starch, so it's comparable to a domestic potato. And there's that tunic tissue that gets removed. Now, the analysis shows that we can do the experiment with and without the stone as a weight. And without the stone, you have to really use your muscles to hammer the ground with the stick. And so using the stone is actually beneficial. So yeah, you pay a cost to lift the stick and the stone against the force of gravity, but you make up that cost with increased force every time you hammer. So the overall efficiency of using a stone is 7 times greater than not using a stone. And I tell you, this project only came to me because I moved to Vermont and I had to start chopping wood. And I went to Dan & Witt's in Norwich, and I was asking, what kind of axe do I need? And I didn't know, and I got like an axe, like a thin axe. And they said, no, you need like the heavy axe with the triangular head. And I thought it was crazy. And then they worked with me. There's 8-pound weights and 10-pound. And they worked with me to figure out the ideal one. Because yeah, you lift it up and you pay a cost to lift that mass. But then you just relax your arms and you let gravity and the mass do all the work for you. So it's more efficient to have a heavier axe when you're chopping wood. And so it's the same principle here. And so we think we figured it out. By doing this experiment, by working with local South Africans, we know exactly why these board stones were made. Yeah, it took a lot of time to make them, and you lug them around the landscape, but you make it up. You make up that energetic cost with improved forging efficiency. Okay. So there, we've given you a little glimpse of our research interests. We have a shared interest in South Africa. Those are complementary interests. Diet, foraging behavior, complements locomotion. You have to move efficiently on the landscape to acquire food resources. So these are related concepts. And these concepts tell us a lot about human origins and human behavior. And South Africa is just great for giving students that same experience that we had when we were undergrads. We were fortunate to have advisors that took us to the field, exposed us to field work, and for the first time we realized that professors had these interesting lives outside the classroom. And Dartmouth is the perfect place to start to give these same opportunities to undergraduate students. So right as Jerry came to Dartmouth in 2015, remember the fall quarter shifted forward in time. So now the fall quarter ends right at Thanksgiving, which leaves this little window of time between Thanksgiving and Christmas. And students are now calling that window the winterim. And so now faculty are realizing there's an opportunity here to extend an ordinary course. So we conceived of a typical fall quarter course that happened on campus, and then there's this 3-week extension that happened in South Africa. And so students could then go to see the areas that inform our own research and that inform paleoanthropology, and honestly, there's just no substitute for seeing it yourself. Now, Dartmouth, with support from President Hanlon, with support from the DCAL, the Dartmouth Center for the Advancement of Learning, and because of the infrastructure that we have, the Guarini Institute for International Education, we were able to conceive of an opportunity to go to South Africa with these resources in hand. And I don't know any other institution that would have precisely this perfect storm of opportunity and faculty interest to do something like this. So our class occurred in Dartmouth Hall right here. We spent the fall quarter reading the literature associated with each of these different concepts in South Africa. And so each week was devoted to a particular topic that would then inform and enhance their understanding of that topic and those materials when we got on site. So we arrived initially in Johannesburg. And Jerry, you should just jump in anytime you want.
Sure.
Okay, all right.
Do it, yeah.
And the beauty of the fossil resources in Johannesburg is that they show the entirety of the fossil record in South Africa, from the Triassic all the way up to modern times. And so the students start to appreciate the magnitude of this time depth with the kinds of fossils available at the University of Witwatersrand.
So then the students got to see the fossils that we had found at Malapa, and many of them had designed their own projects around these fossils. One of the requirements that we had in this class was that the students had to design their own research project. We had 15 students. They designed their own research project that couldn't be answered unless they went to South Africa and collected some data and measured something or asked people something that we didn't know the answer to and couldn't find the answer to if you just kept reading papers, kept reading books. That we didn't have the answer. You had to go and do the research yourself. And so several of the students worked on the fossils that are shown here. This is Karabo. This is the young male Australopithecus sediba. And there's nothing like seeing the original fossils to see the kind of anatomical detail that many of them had their questions centered around. And I can't even tell you, to sort of mirror what Nate said earlier, I can't even tell you how many times I've been in that room working on fossils, and I wish my students were with me, where I could show them stuff and say, hey, God, check this out. Check out this anatomy. This is what we were talking about in class. But they're not there. Or I'm at Malapa, and I'm digging, and I'm working at the site, and we're finding things or not finding things, this is often the case, and I wish my students were there. Or I'm in the classroom, and I'm talking to them about the work I do, and I wish I could just bring them suddenly to South Africa, and we could start seeing the original fossils or going to the site. And this class allowed us to do that. So Katie and Lauren, who showed up a second ago, they did their project on that skeleton. So that image is capturing them talking about breaks on the skeleton that help us understand how these individuals died. What they were interested in was how did these individuals die? And what evidence do we have for cause of death 2 million years ago? Michael and Erica looking at a skeleton of a baboon in this particular image in the museum. So the museum was this wonderful place, or the university museum was this wonderful place where the students could see the fossils themselves and handle them and get a sense of what kind of research happens once you get the thing out of the ground. But then we went to the cradle, and we went to the site of Malapa to dig ourselves. And I got to tell you that one of the intentions of going to the cradle of humankind and going to Malapa and having the students dig at Malapa was to teach them how difficult paleontology is. So many students want to become paleontologists. And we said, oh yeah, do you want to stay in a tent for 5 days where there are bugs flying through at night that are bigger than your hand? And some of them freaked out at that a little bit. That we could hear hyenas at night. The hyenas were very vocal at night, and that upset a few folks. But the landscape itself allowed us, or gave us, an appreciation of not only what the hominids were facing when they were alive, but how the landscape has changed over time as well, because some of the fossils we're finding were of creatures that simply aren't there anymore. But honestly, when we got to the site of Malapa and started digging, and we dug for 3.5 days, the intention, and this is a little cruel, but the intention was for them to fail. I didn't want them to find fossils necessarily. I wanted them to see how hard this is. And you have to dig and dig and dig and dig and dig and dig, and very often you find absolutely nothing. And we showed them that this is a fossil-rich site, and how in certain spots of the rock where her skeleton was discovered, we still have parts of her in there. We can still see those fossils, so we have some targeted areas of excavation. Here's Kathy using some of the equipment that we use at the site to document exact placement of the fossils. When you find something, we need to know the exact GPS coordinates of that fossil. We worked in these gridded off squares where the students for 3 days just had to slowly scrape dirt and sift it into, or shovel it into buckets that then went into sifts, and they shook and shook and shook and shook. And I don't know if you'd agree with this, Nate, but many of them, I think, were bored to tears of this after a day or two, and I was like, excellent, great, you're learning how this works. And then, of course, on day 3, they find a fossil. And they not only find a fossil, right, you know, it's got to be an antelope, right? That's all we ever find. They found a piece of her pelvis. They found a piece of the female Australopithecus sediba pelvis, and it is a critical piece because it helps us understand what the pelvic shape was like, and the pelvis is really important for bipedal locomotion, but also because we think this is a female, this is going to help us reconstruct what childbirth was like, what the mechanics of childbirth were like in something that lived 2 million years ago. Because childbirth in humans today involves rotation, the baby's often facing backwards, and childbirth is often a social event in humans. Whereas in chimpanzees, they go off into a tree by themselves and give birth to a baby that's facing forward. They can deliver a baby themselves, whereas in humans it can be much more difficult than that. In Australopithecus sediba, we're not sure. And having this piece now allows us to ask that question that we weren't able to ask before. And then we went to other sites within the cradle. This is the site of Sturkfontein, where the timing was perfect. We did coordinate this to meet up with an alumni group that was traveling through South Africa at the time. And so our students got to interact with a group of Dartmouth alumni this is the cave of Sturkfontein, where many famous fossils were discovered. And without us even needing to prompt this, it was just this really wonderful moment for Nate and I as teachers that we walked into a museum that preceded the tour of the Sturkfontein cave, and there were fossil replicas all over the place, and our students became the teachers. And our students started teaching these Dartmouth alumni about what they had been learning about, how they would see certain displays and say, well, that's not quite accurate anymore because new discoveries have shown this, 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 and this. And we were able to just step back into the shadows and let our students become the teachers. And it made us incredibly proud of how they took ownership over this experience at Sturkfontein. So we spent a good 5 days in the cradle of humankind, and then to really flesh out what the landscape would have been like of Australopithecus sediba 2 million years ago, we needed to travel to a national park to see some of the animals that would have been coexisting on the landscape, or at least the modern descendants of those animals 2 million years after the existence of an Australopithecus. Nate, I don't know if you want to jump in and talk about Pilanesberg at all.
Sure. Well, one of the common refrains we heard from the students was just how alarming and unnerving and unsettling it is to move through this landscape with these large animals on it, because of course when you have a complete assemblage of herbivores, you also have a complete assemblage of predators. And so I think there was just no substitute for this kind of experience for the students to realize that, yeah, being bipedalism makes you efficient, but it also makes you slow, and there is no way we can outrun a potential predator. And so the students got to see these predators firsthand, and they actually saw a predation event. So here's a lion with a small impala, and the poor thing was just bleeding and calling while the lion commenced eating it. It was pretty arresting for the students. But nonetheless, this is Katie. She's a 19, right?
Yes. Yep.
She wanted this photo in the presentation because she felt like this look on her face captures how she looked all the time during this course. She just so enjoyed it. But we also got together at night around the campfire, and other students did their projects on each other. So there was an engineering student who was using these portable blood pressure monitors, because it's been argued one of the benefits of evolving controlled use of fire is not only for cooking, but it also reduces your blood pressure, it relaxes you, and that could be a selective advantage too. And so sure enough, her analysis showed that these students were getting more relaxed as they were watching the fire. And it's thought that there's something about the rhythm, the fluidity of the flicker of the fire that maybe is very calming and mesmerizing. And then from Pielensburg, we moved to a site called Wonderwerk, rather.
Where there's the first evidence of fire. So we talked all about fire in the class, and then we had a student who was doing a project on fire, and then we said, hey, let's go to where we have the earliest evidence of controlled fire in the human fossil record. And it happens to be at this site of Wonderwerk Cave in central South Africa. We got a little lost getting there, but we eventually did get there. And down in those deposits is a layer of ash, and it's been dated to 1 million years old. So long after Australopithecus, but long before us, presumably something like a Homo erectus had controlled fire. It wasn't yet making fire, we don't think, but it was controlling natural fire, and it was using it to cook its food and perhaps to sit around and tell stories at night and lower its blood pressure the same way we do today. And in that same cave, the students noticed rock art on that cave wall as well, which has been dated to, I believe, early Holocene, late Pleistocene, early Holocene. And so this is a cave that's been occupied for the last 1 million years, and perhaps even further back than that as they dig down and find older and older and older spots.
And then we had an opportunity to meet the makers, presumed makers of that rock art, a population of indigenous peoples called the San that live in this northern part of South Africa in the Kalahari Desert. So this was a radically different habitat from what they'd experienced before. This was a true, proper desert habitat. And this particular population has sovereignty over an area of a trans-frontier national park, which means that the park goes into different countries. And they built a lodge on this site, and so we thought it was important for the students to interact with the San as much as they could, so we visited this particular lodge. And this entailed working closely with the San to go on nature walks, to see the different kinds of plants and animals that were important to their survival and ecology in this particular habitat. Some of the students decided to do projects with the San to wrestle with this tension that in anthropology is called essentialism. What is your innate behavior, and to what extent do you change your innate behaviors in association with others? And so they were interested with how the San navigate this complexity of how their essential behaviors are changed when outside groups try to commodify or commercialize their behaviors for their own purposes. And so the students worked directly with the San to ask these kinds of complex questions. And here's an example of one of their informants, another informant here. And the students it was such a moving and profound experience for them, because the San said that no American students had ever come and asked these kinds of questions. And some of our students were native themselves, and they felt a strong just camaraderie that I think can't be replicated by reading the literature. The academic literature that we assigned in class has a dry, muted tone. It doesn't have any of these more emotional, more complex elements that the students experienced firsthand. And on our way to Cape Town, they met another individual of San descent. He was easily recognizable. He was a waiter at this one, I don't know, it was like almost like a roadside attraction type of deal. And they pulled him aside and they asked him similar kinds of questions, and they got very similar kinds of responses. And so the students recognized that the indigenous identity and indigenous thoughts are not necessarily being filtered into popular media and in the academic literature. So for the first time, the students realized that they were learning something new and different, that they were directly understanding a situation that wasn't necessarily being portrayed that way through other sources. And there's no way to get that experience without going yourself. So from the Kalahari, we shot way down to Cape Town, and I don't know.
It was a huge change of scenery to go from the Kalahari Desert to Cape Town in 24 hours. And the students, one of the things that was important to us was to make sure that the students had an appreciation for being in South Africa, not just for the science that we do and the science that has been done for decades, but to understand that science is not done in a vacuum, and that science is always done in a world or in a socio-political climate that has an impact on those scientists. And so we wanted them to understand to some degree, this wasn't a history class, this wasn't a political science class, but we wanted them to understand the history of South Africa. And we wanted them to understand apartheid and the effect that that had on South Africa and South African scientists and continues to today. So the students, we went to Robben Island, and we spent time in Robben Island learning from one of the prisoners himself that talked about the experience of being in Robben Island with Nelson Mandela. That's Nelson Mandela's cell over there on the right. And it was an incredibly moving experience for all of us to be there and to think about, again, how the science is done today in this country that is changing so rapidly politically, and how it was done in the past. I think about a colleague of mine, Philip Tobias, who was in South Africa during apartheid and fought the regime from within. So he had enough power that he was able to make some changes so that black students could be admitted to the medical school during the apartheid regime. And so when apartheid finally fell, he was seen as a national hero. And this was a paleoanthropologist. This was somebody that I saw as a scientific hero, but he's more than that. And we were able to appreciate that to a much more degree, I think, by spending time in Cape Town, spending time on Robben Island, and then eventually, of course, going across to Table Mountain, where the students got all geared up to climb Table Mountain, right?
Yeah. I don't know what they're doing here. This photograph has meaning to them, but they're excited because it's the only place in the world where you can see one UNESCO World Heritage Site from another UNESCO World Heritage Site. So the Robben Island is a World Heritage Site, and Table Mountain is one. So I'm not sure exactly what they're doing here.
Are they spelling something out?
What?
They're spelling something out, maybe.
Oh, are they?
They're sending us a code.
Okay, I didn't realize that. Okay, all right. And then, of course, they wanted to climb Table Mountain, so this is their, they're looking really fierce and ready, and they're ready to tackle this mountain. So.
And they did, and they're in much better shape than I am, it turns out, because I went with them and it was painful.
And in addition to seeing these cultural monuments, they also went to the coastline to explore how baboons consume coastal resources, because an emerging thought in anthropology is that there's something about the South African coastline, because you have rich marine resources. Formerly, about 200,000 years ago, the sea levels were much lower, and there was this broad savanna. So there was an area of land about the size of Israel or New Jersey, whatever your frame of reference might be, that's now submerged. And that was flat. It was a savanna. And so the idea was that in addition to underground storage organs, reliable sugar, in addition to the marine foods and savanna resources, reliable protein, that this was a perfect storm, a trifecta of resources that allowed humans to forage especially efficiently. So there's something really special about the South African coastline. And one particular student wanted to do a project on baboon-human interactions. And so I just want to show you very, very briefly what a final project looks like at Dartmouth now. So you may be familiar with term papers, but here so in this particular class, we were more focused on term projects that would have an online capability. And some students really took this very far, like her project entails maps where she interviewed subjects about their views on baboons. It's color-coded based on how tolerant they are to baboons. She has all sorts of data on perceptions and human-baboon interactions. So she compiles all this into a website that is then turned into us. And then there's a project reflection piece where they think about their experience and think about how the experience transformed their conceptualization of the material. So we're trying to move away from the term paper and provide a medium that allows students to expand their conceptualization of particular topics in a course as a whole.
Yeah, and I'll add to that, Nate, that one of the things that we wanted them to be able to do is feel able to fail. We wanted them to be able to go to South Africa and realize that the projects they'd come up with were untenable, or that the hypothesis they had was actually wrong, and that the data that they were collecting was either different than they expected or so much more complex than they once thought that their original project just couldn't be done. And over the course of the 3 weeks we were there, many students expressed frustration to us because these are Dartmouth students. They've never failed before. They're good at everything. And they came in and they said, oh, my project is falling apart. And we're thinking in the back of our heads, good. Your project is falling apart. You're learning. Now pick yourself back up and turn it into something. How are you going to now turn this into something different? How are you going to reflect back on this experience and have it change you and change how you would tackle a project next? And we'll see some in just a second.
And they were resilient. They really were, yeah. Which was a great experience for us. From Cape Town, we moved to Agulhas Point. So if you can imagine an area about this large that was now submerged, but was formerly a savanna-like habitat. So we wanted students to see the remnants of that now submerged savanna. So we went down to the coastline at this site. This is the point where the Indian Ocean meets the Atlantic Ocean. They were thoroughly impressed with that site. So they got to see both the marine resources, the limpets and the mussels and the clams that are on the coastline, and they could envision the savanna habitat because some of that botanical aspects of that habitat still remain. So they could see it all, and they could start to understand why this habitat may have particular importance. One of the most meaningful events for the students was a group dinner that we had in our house. And I don't know if you want to add to that. They constantly talk about this night as being one of the highlights of the trip.
It was towards the end, and there was a lot of reflection that happened that night of, okay, we're just about done, and now you reflect back on everything that happened, because boy, we really packed it in. Yeah. We did a lot of stuff in those 3 weeks, and it was a moment for them to sort of think back and reflect on that experience, because they knew they'd be leaving soon.
And one of the exciting outcomes of this project, so here's the very last, second to last day, we're at a cave site called Pinnacle Point, which is important because it has some of the earliest evidence for potential language, the earliest evidence for using a spear thrower, so projectiles. So Neanderthals, remember, yeah, they were smart, but they had thrusting spears. And modern humans come into Europe with spears that you can actually throw. So we think it was a big technical advantage. We see the earliest evidence of symbolic culture, engravings, artwork, that sort of thing in South Africa on the coastline. So something exciting is happening on the coastline. Pinnacle Point has the evidence for a lot of those, the emergence of this important kinds of behavior. And so one student really got just turned on by these caves and thinking about the importance of caves in human evolution. And so he's now a presidential scholar, and he's developing his own independent projects looking at this one hominin that has extremely curved fingers. And typically we associate curved fingers with wrapping the fingers around branches. So orangutans and gibbons have very curved fingers. But this hominin has greater finger curvature than even orangutans. And yet the wrist and proportions of the hand are just like ours. So I don't know, it's a big shoulder shrug. What was it doing with his hands? We don't know. And he thinks maybe it was in caves, rock climbing, because he himself is a rock climber. He's the president of the Dartmouth Mountaineering Club. And so here he's trying to unite his passions for climbing with anthropology to explore another project in South Africa.
Another presidential scholar, Olivia Weiner, shown here. She's going to be working with me this summer looking at a lot of the fragmentary fossils that we have from these South African cave sites. Very often they're not found complete, they're found in pieces. And she's going to look at how we can take those pieces and reconstruct them into whole skeletons. So if we had a whole series of skeletons which we do. We have access to those sorts of things. What if you only had a fragment of that bone? Would you be able to reconstruct its entire length? And so she's going to be doing a project like that with me this summer, in part inspired by what she saw in South Africa of all these fragmentary remains and saying, geez, it'd be great if we could squeeze more information out of these fossils. Another presidential scholar from last year, Jessica Kittelberger, who just I think she did 2 different projects, maybe 3 different projects when she was in South Africa. She could not get enough. She's also been working with me on a lesser known idea in our science that brains have actually gotten smaller in the last 30,000 years. Most people think brains have gotten bigger and bigger and bigger and bigger and bigger and bigger, and they haven't. Brains got bigger, and in the last 30,000 years, they're actually heading in the other direction. And they're about 15% smaller than they were 30,000 years ago. We don't know why. We don't know when this started. Lots of folks have their own little jokes on human intelligence today, and it keeps going down, 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 perhaps. I like to think it's actually a result of collective intelligence, that one individual doesn't need to know everything there is to know about being a human anymore for survival because we rely on other individuals. And so we're becoming a little more eusocial. But she realized that there was a huge gap in our understanding of brain size at certain places at certain points. And while we were in South Africa, she took advantage of this and measured the brain size of several skulls from a cave site in South Africa that are about 5,000 years old, which is not very old for a paleoanthropologist, and yet for this particular question, it actually fills in a key time period that we don't know very well for brain evolution. So she's been working on that particular project.
So I think I'll just take a minute to summarize, then you should add something.
Sure.
Just to say that in some respects, I feel like this presentation to you is a bit premature because what's so exciting about these students is that they're now doing their own individual projects inspired by the content of the class. So I just can't wait for a year from now or 2 years from now when the true arc of the outcome will be revealed. And Michael Everett is so funny because he came into your office and he said, you know, I don't see the need for any more classes. I just want to stop taking classes and go to South Africa and do as much work as I can. And some part of me felt like, that was just a terrific outcome.
And he's a second-year student. It's not like he's a senior. No, I totally agree. And not only a year from now or 2 years from now, I want to know 20 years from now what kind of experience what kind of impact this experience had on their educational trajectory. So when they come back for reunions, 20 years from now or so, I hope we're still here doing some sort of talk on our research, and I want to know how that impacted them, because it impacted us enormously. It totally changes how I think about teaching in the classroom and how I can use, maybe not take every class to South Africa, but there's got to be a means by which we can incorporate more of this experiential learning approach into all of our classes, given how effective I think it was with this particular group. So, thank you.
We have time for a few questions.
Sure. Great.
Well, first we have microphones, so wait for the microphone so everybody can hear your question. And while Allison's coming down, I just want to mention that Jessica spoke to some of our alumni before the trip, and she was talking about that specific project. She was measuring the capacity of the cranium with like wheat berries or something.
Yeah.
Sorry.
The question, it was about how Jessica was measuring brains. And we were running around South Africa going to grocery store to grocery store looking for millet seed to fill up the skulls. And we ended up using little pieces of rice that did the same thing. So we were able to measure volume that way.
Yeah, can you hear me? My question is this. You're talking about humanoids or whatever, hominids from 2 million years ago. Do you run into students who 40% of the American population thinks people came on the earth 6,000 years ago? Does Dartmouth have students like that? They probably don't take this course. And then how do you contend with that?
So, and then maybe you. So it's unusual. It's unusual to encounter a student like that, but I have. And oftentimes the student comes to me with a legitimate struggle that they're really trying to negotiate these different pieces of information that they're getting in their heads. And I say, hey, that's one of the beauties of being a human and having this brain is to take these different kinds of information that you're getting and rattle it around in there and come up with your own ideas. However, my job as an instructor in biological anthropology is to teach people about what science is, how science works, what the nature of evidence is, what we know about our ancestry based on these methods. And so if they leave the class still not believing in evolution, I don't really care. To me, it's not about convincing anyone. It's about making sure that they know at least how we do our work. And if they can then go out into the world and know at least what we do and how we do it and not spread misconceptions about what we do, then I think we've done our science a service. But I don't think we're at least I'm not in the position of trying to convince anyone. I think if we present them with the evidence we have and we give them experiences like this and show them, look, these things are real, they existed, and we have the physical evidence of them, and here's how we date them. We don't just make up dates. We understand the geology and the chemistry of the rock, and here's where the dates come from. If they can understand that more and more and more, then I think that they'll realize that any rejection of this idea kind of flies in the face of the evidence that we have.
That was good.
Oh, good.
A question for Dr. Dominy. Do you know the dating of your phalangeal hominin, or the curvature hominin phalanges, and what date and where this was found?
Oh, the hominin with the curved phalanges is called. The question was about the particular hominin that I mentioned that was the focus of study by Michael Everett, a 19. He's interested in a hominin called Homo naledi. It was recently discovered. Jerry's a co-author on those papers, so he's better suited than me to talk about it. The date was just published about 2 weeks ago, and it's about 200,000 years old. So this Homo naledi lived on the landscape at the same time as us, our species. So there were at least two versions of Homo living in the same place at the same time, one with extremely curved fingers and one with fingers like ours. So presumably that curvature means something biologically. It was doing something with its hands. Personally, I think it was digging, right? And without a bored stone. Think about it. If you have to dig with a stick with no extra weight, you have to grip that stick very hard to dig in that rocky soil. So I kind of think maybe it was a digging adaptation, but I don't know. But Michael thinks it's rock climbing, so.
Well, we'll see. And we all thought it was tree climbing. When we first found it, we all thought it was, okay, curved fingers climbing trees. That's what curved fingers tell us in the fossil record. But that's what is important about having collaborators like Nate is that, I mentioned to him, oh, it's climbing trees. And you say, not if it's 200,000 years old, there are no trees. Or the trees are not, they're acacia trees, and who wants to go up in those? So knowing the landscape at that time and working with somebody who understands, landscape ecology, it becomes incredibly valuable to paleontologists trying to reconstruct what these creatures were doing.
So the landscape you saw was fundamentally the same 200,000 years ago, mostly acacia trees. Acacia trees produce pods. That's not something a human would eat normally. So there's no ecological incentive to climb a tree to consume those things. And besides, trees are relatively sparse in the landscape. So it's that kind of thinking that led Michael to think creatively about what other kinds of things could this thing have been doing. Rock climbing maybe is one of them. Golf, oh, yes. Let's see, the hip swing would be your area.
Ah, there we go. Yeah. We'd have to look at it. Does have some interesting rotation going on. Yeah, all right.
As a one-time Dartmouth cross-country and track runner, I was fascinated by your work in locomotion, and it appeared to me from the animated simulation that you showed us that the subject there was moving really flat-footed, couldn't see much flexibility in the foot, and as a consequence must have been very slow, a lot slower than a Dartmouth runner.
It's a great question. We don't have her complete foot. So there are lots of pieces of her foot that remain a mystery, and I can't wait for us to get more of her foot anatomy. But what we do have from her would suggest that she is more flat-footed than the majority of humans today. But compared to a chimpanzee, she would have a partial arch. So her foot would be somewhat arched compared to a chimp. But compared to most humans today, we would say she's definitively flat-footed, and even more flat-footed than folks that consider themselves to be flat-footed. Oftentimes, the architecture of your bony anatomy in there is still kind of curved, and it's soft tissue that is producing that flat foot. However, with her midfoot, in the middle of her foot, and the animation was probably moving too quickly to see this, we're going to produce another version that slows her down. She does have a bend in her foot. In the middle portion of her foot called a mid-tarsal break, which most humans do not produce. Chimpanzees do, other primates do. When they lift their heel, the middle portion of their foot actually bends in half, and then they roll onto the sole of their foot and their toes bend. Whereas most humans lift their heel and their foot is quite rigid, in part because of the arch, and all the soft tissues make the foot rigid. And when you lift your heel, it works as a lever, an effective lever, that raises the foot onto the ball, which then you can push off and propel yourself into the next step. There are some humans who do produce a mid-tarsal break. And we discovered this somewhat accidentally collecting data at the Boston Museum of Science on a bunch of visitors who would walk across a planar pressure device and produce a digital footprint. And I would see these footprints, and they looked like, in some ways, looked like ape feet. They had this mid-tarsal break. And I'd ask the people to do it again. Can you walk again? And they'd do it again, and they'd produce another one of these. And I'd say, this is awesome. This is no one knew humans could do this. And it turns out a good percentage of the human population can do this. They have weak enough feet that they produce some floppiness in the middle portion of their foot. So we MRI'd a bunch of those people. And it turns out their bony anatomy, the bones of their feet, tell us they can do this. And that anatomy matches what we see in this skeleton. So we think this skeleton also had that flexible midfoot, which would have been great for tree climbing, would have been really beneficial for getting up that tree, grabbing onto that tree, which we think these things were still doing, but it comes at the expense of how well it could walk. And most likely, I agree with you, most likely on that flat foot that's very floppy, this thing would not be able to walk as efficiently as we do today, or as quickly as you phrased it. Mm-hmm.
I'd like to ask. Oh. Sorry, just one more. Okay. I'd like to ask about the level of what kind of scholarship that you have in the scientific community in South Africa, and did you interact with them, and what were they doing? I asked because I was bureau chief for the New York Times for 4 years when Mandela got out of jail. So, and I never heard of any of this. Didn't have time. It's fascinating.
We made a very concerted effort that at each location we wanted to work with local South Africans and interact with their students. So at each stop on this extension, this embedded course, we were interacting with students. And in Cape Town, I remember vividly, we went to dinner with faculty and their students. And the nature of the conversations just went in all directions. So yeah, for us it was a top priority to reach out to our colleagues there and students there, and to make sure that our Dartmouth students could interact with South African students.
Okay, well, if you could all...
One more.
Oh, was there one more?
Yeah, one more.
If you guys are game
What,
for it.
I'm game.
Is this turned on?
Yeah.
Okay. My question is, why is it that South Africa happens to be the so-called cradle of humankind? Is it because of lack of volcanic activity? Because there might be other places in the world where there were even earlier records that were destroyed by metamorphism.
Sure, it's a great question. So I think each country where early human fossils have been found has laid some claim to being the cradle of humankind. And in South Africa, that region that UNESCO World Heritage Site happens to be called the Cradle of Humankind, but I don't think any of us would necessarily make the case that it was South Africa, where all of this was happening. Instead, what South Africa does is it produces this window of these different time periods where we see some key innovations in human evolution happening. We even talked about, could we have done the same kind of course in Tanzania? Could we have done it in Kenya, in Ethiopia? And the answer is yes. We could have done this course in those countries as well. We could have done it in Uganda. And in each of those places, there would be this special kind of unique flavor that each country provides to our view of human origins. But we couldn't have done a human origins class outside of Africa. Africa is where our lineage evolved. It's where the split with chimpanzees, the very earliest hominins, were in Africa. We know our genus Homo evolved in Africa, and we have very good evidence that the species Homo sapiens evolved in Africa as well. So any class that purports to look at human origins is going to Africa. It's just a matter of which country it is. And South Africa, for us, given the research we do, was the obvious fit. But I could see this, kind of class happening in Kenya.
Okay, well, great. I'm sure you can all see why I enjoy working with Professor DeSilva and Dominy so much. Their enthusiasm and their scholarship and dedication to the students is really unparalleled. And I thank you and invite everybody else to thank you as well.